So my name is Chris Ching. I'm the co-chair of the ENIGMA Bipolar Disorder Working Group here at the Imaging Genetics Center, part of the Keck School of Medicine at the University of Southern California. And today we're going to be having three brief presentations about some ongoing work that's happening in the ENIGMA Bipolar Disorder Working Group. I'll start with an overview of the working group, what we've been doing over this past decade, and looking forward to the next decade of studies. And then we'll hear two other presentations focused on modeling medications in bipolar disorder. I like to start with some acknowledgements. These studies are not possible without the thousands of study participants from around the world, the over 200 ENIGMA Bipolar Working Group scientists who make all of this work possible. So what I'm going to be talking about today is how the ENIGMA Bipolar Disorder Working Group is combining existing neuroimaging samples from around the world to improve replication and generalizability of findings in bipolar disorder. I'll give you some recent study highlights, including how these studies are motivating future goals that move beyond cross-sectional and case versus control type analyses. And I'll also talk a little bit about some efforts underway to collect deeper phenotyping or more detailed clinical and demographic information and applying novel modeling techniques to try to contextualize the neuroimaging signatures that we see in bipolar disorder across different biological levels, as well as across disorders. Now, the ENIGMA Consortium is a global network of over 2,000 scientists across many institutions and countries, combining existing neuroimaging data from many thousands of individuals and studying a range of brain conditions. Now, the bedrock of the ENIGMA approach is to really try to take these diverse neuroimaging samples and apply standardized processing and analysis methods, sort of like cookbook recipes, to extract standardized brain measures across these different data samples. And so we have over 50 different working groups, some of them focused on developing those standardized processing and analysis methods and algorithmic development. And then, of course, we have the other working groups focused on a range of brain conditions, including our bipolar disorder working group, which we established in 2012. It includes over 50 international cohorts, including over 200 scientists from 20 countries, pooling data from 3,500 individuals with bipolar disorder and close to 9,000 undiagnosed healthy controls. And again, I co-chair the group with my colleague, Dr. Ulla Andreasen from the University of Oslo. So how do these studies work? So when combining existing data samples, the ENIGMA approach really is about being very pragmatic. And so we know there's lots of different privacy and local ethics boards considerations when thinking about pooling these types of data. And so the first sort of important principle is that you don't need to share any primary data to participate in these studies. And so in many cases, we show people how to run these standardized processing and analysis protocols so that we can combine data either meta or mega analytically. Each site always retains complete control over their data. They determine which projects they'd like to participate in. And so these studies really operate on this opt-in principle where we're trying to build the largest studies, but being pragmatic about how we can combine different types of data. So the two major sort of goals and guiding principles of the ENIGMA Bipolar Group are looking at whether we can provide more reliable discoveries that improve our understanding of the pathophysiological mechanisms in bipolar disorder, and really looking to see whether we can provide more clinically relevant information to improve pathology, diagnosis, mechanistically directed interventions, and hopefully improve treatment outcomes. So if people are interested in more details of what we've been doing over the past decade in the working group, I encourage you to take a look at a recent review article that we published, provide a lot more detail than I can provide here today. Now, since 2012, we've published 15 studies. Each of them is the largest ever of their kind. We have about 18 ongoing projects. You can see a full list of those in details on our website. But generally what we've been finding is that, and this is the most important thing, bipolar involves subtle alterations to brain structure. So we're seeing smaller, thinner gray matter regions, altered white matter integrity. We see these differential effects of commonly prescribed medications. We've shown some modest predictions using AI and machine learning techniques. And we're also performing some of the largest, really the largest cross-disorder brain studies, which I'll talk a little bit more about later. Now, in terms of the decade ahead, we're really trying to focus on more finely mapping some of these brain alterations that we've shown in bipolar. So using more advanced brain morphometric features, including vertex and voxel-wise analysis, improving classification and individual level prediction using data-driven and machine learning techniques, really trying to move towards modeling how brain change over time is related to illness progression. I'll talk about that a bit more in a moment. Trying to investigate the impact of polygenic risk and gene expression markers on large-scale standardized brain measures. We want to see how individual symptoms, functional domains, risk factors of the sort map onto these measures, these standardized metrics of brain structure and function, not just within bipolar disorder, but across diagnostic boundaries. And of course, we're also really trying to empower future mega-analyses that allow for more sophisticated study designs. And so in this past year, we published the largest longitudinal study of brain change in bipolar disorder. If you're familiar with some of our studies, the sample size here, you'll notice, is quite modest compared to some of our past studies, including thousands of participants. And that's because there's really limited longitudinal data in bipolar disorder where people or individuals have been followed over time. And so again, still the largest study of its kind. What we found were these really sort of subtle alterations over a yearly rate of change in patients versus controls. In some cases, the controls or the undiagnosed individuals were actually changing more than the patients. So again, these subtle alterations over time, we're going to keep expanding this analysis to look beyond sort of a year. And then I think importantly, we also showed this, the number of hypo or manic episodes was associated with faster cortical thinning. Again, pointing to this idea that controlling symptoms may be a factor in improving long-term brain health. Now, one of the other things we're looking at is whether potentially modifiable clinical factors like obesity, here measured using body mass index, influence the way we understand bipolar-related brain signatures. And so we recently published, again, the largest study of its kind looking at body mass index. We found that individuals with bipolar tended to have higher BMI compared to controls. And this is for a number of well-documented clinical factors, including the medications people are taking, which can result in higher BMI. And what we found, interestingly, was that higher BMI was associated with similar brain region alterations as we see in bipolar, but importantly, it may account for the extent of those or the severity of those brain alterations. So pointing to this idea that it's really important that we model these complex clinical factors to understand how large or small these effects are in the brain. Now, as I mentioned before, one of the steps that we're taking to try to improve our modeling is to use more high resolution brain shape features. Here's a deep learning framework in which we're trying to use those high res features to improve diagnostic classification, right? Can we do a better job of telling who has the illness from who doesn't, but more importantly, trying to chase down some more clinically relevant classification. So really trying to predict things like severity of illness, symptom profiles, the types of medications people are taking, and how they're responding to those medications. And now, one of the things that's happened over this past decade of ENIGMA studies is that we're beginning to realize one of the initial goals of the ENIGMA Consortium, and that's to be able to directly compare brain signatures across different illnesses. And so each of these brains represents the largest case versus control cortical thickness study of its kind that's been published in these different illnesses. And so one of the things we're doing now is taking these sort of unprecedented sample sizes, each of these studies, right, using the same sort of cookbook recipe to derive these brain features and directly compare the brains across disorders. And so this is an effort that's underway now that we're leading, looking at combining data from the ENIGMA Bipolar, Schizophrenia, and Major Depression working groups. So we know these conditions have overlapping symptom profiles, responses to different medications, overlapping genetic risk profiles. And so what we're doing here is really to try to look at directly comparing these brain measures to find overlapping and distinct brain patterns across these behaviorally defined disorders. Now, one of the important steps we're taking also is looking at when we ignore the diagnosis, right? So we lump all the data in together. These large samples may provide us an opportunity to resolve alternative patient groupings based on dimensional measures such as symptom profiles, treatment response, duration and severity of illness that spans these classic diagnostic boundaries. Now, in an effort to look across disorders and relate what we see on an MRI level to underlying genetic risk, an early study that we did, a recent study that we did, was we looked at how the similarity in brain signatures across these large-scale ENIGMA studies to some extent seems to mirror the underlying genetic similarities we see from large genome-wide association studies. Now, this is important because we know that many of these conditions have a strong genetic component. And so here we're trying to draw a better link between what we see on an MRI level to the overlapping and distinct genetic profiles. Now, another way of doing this, Thomas Paus and Yash Patel have been also conducting here, published two large-scale cross-disorder studies from the ENIGMA Consortium, looking at whether MRI-derived case versus control cortical thickness and surface area differences might be related to underlying profiles of cell-specific gene expression using tools like the Allen Brain Atlas and PsychEncode. And what they're seeing here is that there's this potential shared and distinct neurobiology underlying these cortical alterations that span these psychiatric disorders. And they point to some interesting, future potential hypotheses regarding the mechanisms that might be driving risk for the development of these mental illnesses. Now, as I mentioned before, we've been modeling the ways in which medications affect brain structure and function. And I said before that we've seen how common medications prescribed for bipolar patients can differentially affect brain structure. And that complicates our understanding of illness-specific brain alterations or biomarkers in bipolar. We also know that individuals with bipolar disorder are rarely on a single drug or monotherapy. And so we really need more sophisticated modeling of the polypharmacy or multiple drug interactions that influence some of these brain metrics. And so what we've been doing in this past decade really is looking at taking a pragmatic approach to modeling medication. It's the classic way people model it is they lump drugs into these indication-based categories. So there are many different types of drugs that could be considered anti-epileptic or antipsychotic. And generally what we see is that lithium tends to make brain structures larger, right? So larger subcortical volumes, thicker cortex, less disrupted white matter. And we sort of see the opposite effect when you look at anti-epileptic and antipsychotic drugs. Now, again, we see some differences where typical or first generation antipsychotics, we've seen greater cortical surface area, but generally we're seeing this push and pull or differential effects on the brain structure when people are taking different medications. Now, what we really should be doing and what we're doing now as part of what we call the ENIGMA Bipolar Medications Initiative is trying to delve a little bit deeper into these medication effects. So we're trying to standardize even deeper phenotyping or more detailed clinical, demographic, behavioral measures, along with detailed medication information like dose, duration, serum level, to not just model drugs based on their indication or these classic sort of grouped categories, but really look at modeling medication based on the underlying pharmacological mechanism. And that's what we're doing now. And then in the two presentations to come, you'll hear a bit more about some of these machine learning and data-driven techniques that we're using to try to disentangle some of these polypharmacy effects from some of the complex clinical and demographic factors. Now, in closing, I'd like to just come back to the ENIGMA Bipolar Disorder Working Group map. I think we're quite proud of the effort that we've put forth over this past decade to combine really the most diverse neuroimaging samples to study bipolar disorder to date, but we really have to be honest about the glaring omissions that we see in this map. And while we probably won't ever fill in this map completely with MRI samples, as we continue to try to drive the most replicable and generalizable results, we have to think about ways we can supplement this information, fill it in as best we can, And so that the findings that we derive from these large-scale studies really apply to everyone that's affected across the world, the millions of people affected by this illness. And so we always end with an invitation. If you know of samples or studies that haven't been included in this effort thus far, please reach out, and we look forward to speaking with you. Now, again, I just like to come back to acknowledging the thousands of study participants, their families, caretakers involved in all of these different studies, and of course, the ENIGMA Bipolar Working Group scientists who make all of this work possible. And lastly, just acknowledging a couple of key funding sources, including a recent National Institute of Mental Health grant that we received, as well as a Bazuki Brain Research Fund and Milken Institute Center for Strategic Philanthropy grant that we received to jumpstart this medications initiative. Thank you very much.
Hi, everybody, and thanks for tuning in to our group of talks today. We're very excited to be showing you these findings that we've been working on recently. So what I'm here today to talk about is a new ENIGMA Bipolar Disorder Working Group project. Specifically, I'll talk about lithium use in hippocampal volume, recent mega-analysis that we've been working on of individual level MRI data from the ENIGMA Bipolar Working Group. My name is Sinead King. I'm working at the Centre for Neuroimaging and Cognition, Cognition and Genomics at the School of Medicine, National University of Ireland, Galway. I'm working with Professor Colm MacDonald, who is co-leading this project with Professor Paul Thompson and others. We've had a number of really great funding streams, such as the Milken Institute and the Bazuki Brain Research Fund also, that has allowed us to work on this project, so thank you to them. So just to provide a little background into the project itself, so the use of polypharmacy in the treatment of bipolar disorder complicates our understanding of illness-specific brain alterations and their associations with treatment response. So, for example, in the literature, it shows that lithium in general is associated with increased hippocampal volumes, so subcortical regional brain volumes, and other medications such as anti-epileptics. It's the results in literature is a little bit more mixed, but it's suggesting that these drugs are sometimes associated with reduced brain volume. And of course, when investigating the relationship between medications and brain structure, you also have to consider the differences in underlying pathology also. So to fully interpret the contribution of neuroimaging findings to our understanding of bipolar disorder, it is important to establish what the interaction is between brain changes related to illness pathology and those due to drugs and their interactions, and ultimately what the changes are in relation to these interactions and what they represent in relation to treatment response. So that's what the main aim of the ENIGMA Bipolar Disorder Working Group's project that I'm going to be talking about today is about. It's called the ENIGMA Bipolar Disorder Working Group Medication and Clinical Phenotypes Project, where we continue to map polypharmacy effects in bipolar disorder while accounting for and co-varying for detailed individual level data. And this is the largest mega analysis of its kind. So whereas prior studies were able to use meta-analyses to investigate particular medications and their associations with brain volume, this mega-analysis goes that step further and will instead allow assessment of multiple medications and their associations with the brain at the same time. So we then continued to collect data from 27 sites. Altogether, subcortical volumes were computed from using the ENIGMA standardized protocol of T1 weighted MRI scans. And we performed a pooled mega-analysis comparing subcortical volumes between patients with bipolar disorder and controls, and then associations in patients with the lithium treatment. Analyses were carried out using linear mixed models and moderation analysis, while adjusting for a range of covariates, including other medications taken. along with the important key covariates as usual, such as age, sex, and intracranial volume, as well as the scan site. So in total, there were 4,863 subjects in the data set, 2,231 bipolar patients, and 2,631 controls. So from these, 2,141 were male, so around 44% of the sample, and 2,723 were female. So in patients, we had a number of data for bipolar subtypes. So 1,394 patients were categorized as bipolar disorder type 1, 476 with bipolar 2, and 102 patients with bipolar disorder not otherwise specified. So then comparing between patients and controls, you can see a significant difference in both BMI and age at time of scan, which was increased in patients compared to controls, and full scale IQ was significantly lower in patients compared to controls. We then compared patients taking lithium versus not taking lithium. We saw that patients not taking lithium were significantly younger than those that were taking lithium. We also find differences in clinical and illness severity measures in patients taking lithium versus not taking lithium, in that lower illness severity, such as lower total number of hypomanic episodes, lower total depressive episodes, and lower total mixed episodes in patients on lithium versus those not taking lithium. And again, patients on lithium tended to have lower depression severity based on using the Beck's depression inventory. Then separate to that, to compare patients taking an anti-epileptic versus not taking an anti-epileptic, we see again significant differences in a number of variables between those patient groups. So in those not taking an anti-epileptic, patients had a lower BMI and were significantly younger. Again, lower IQ score in patients on an anti-epileptic versus not. And patients on, again, an anti-epileptic had more severe illness as measured through mood episodes. So they had significantly higher number of manic episodes, significantly higher number of depressive episodes, and they had a more severe depression severity score using all Beck's depression inventory, the Hamilton, and the CSD. Patients on an anti-epileptic tended to have more depression severity scores. Then in terms of medication status in the sample, we had 35% taking lithium, around a similar number of patients also are well on an anti-epileptic, with 7% of the sample on a typical antipsychotic, whereas 34% of the sample were on an atypical antipsychotic, and 28% were taking an antidepressant. And then, so the results of the hippocampal volume comparison, we again showed that larger average hippocampal volumes in patients on lithium compared to those not taking lithium. And this was a comparison after adjusting for scan site, age, sex, intracranial volume, and other medications taken. We found a similar pattern of results when using the thalamus volume comparison, where larger thalamus volumes were found in patients on lithium versus not taking lithium. And this was significant again after correcting for scan site, age, sex, intracranial volume, and other medications taken. Then just moving into our moderation analysis, we found that the effect of lithium on hippocampal volumes was moderated by whether a person was taking an anti-epileptic versus not. We found a very similar pattern of associations, where the effect of lithium on hippocampal volumes was again moderated by atypical antipsychotics. and this finding was not significant with typical antipsychotics or antidepressants, such that typical antipsychotics or antidepressants did not moderate the association between lithium and larger hippocampal volumes. Again, we ran the same analysis using the thalamus and found similar results again, where the association between lithium and larger thalamus volumes was moderated by antiepileptics, and again by atypical antipsychotics. So when you replace the antiepileptic variable with the atypical antipsychotics, you find that again significantly moderated the association between lithium and larger thalamus volumes. And again, this was not significant when replacing the moderating variable with either typical antipsychotics or antidepressants. And so then, just in summary of our preliminary analysis of this very large mega-analysis, smaller hippocampal volumes and thalamus volumes in bipolar disorder appears modified by lithium treatment, even in the context of polypharmacy or other medications taken. In those with bipolar disorder, both antiepileptics and atypical antipsychotics significantly moderated the positive association between lithium use and hippocampal volume, as well as between lithium use and thalamic volume, resulting in smaller brain volumes. Typical antipsychotics and antidepressants did not significantly moderate or interact with lithium's effects on the regional brain volumes studied here. And so future directions, we ongoing multivariate analysis of a wider range of medications and clinical phenotype data will be examined and included in a publication which we are working on at the moment. We also plan to use neuroscience-based nomenclature, which we are working hard on at the moment, and welcome any thoughts, suggestions on using this kind of new strategy of using a mechanism of action approach rather than a typical standard ways of measuring antipsychotics, antidepressants, and lithium, and so on. And we also plan to use more complicated and advanced machine learning methods, and an example of an advanced statistical method called Hydra is a useful tool that might separate clusters of patients in how they differ from controls as a reference population in factors such as X, Y, and Z, where X and Y might be clinical phenotypes and medication data, so that you have clusters of patients that are differed based on those medications and clinical phenotype data, which we plan to incorporate as well. Just want to say thank you to the study participants, because without them, this project would not be possible. And thank you to all of the ENIGMA Bipolar Working Group members. Thank you for listening, and looking forward to hearing your comments, feedback, suggestions, and questions. And just to follow up briefly, I'll show you this neuroscience-based nomenclature where we have got to so far with this NBN approach. So whereas you have these traditional categories of antipsychotics, antidepressants, and antiepileptics, we have instead converted them using NBN categories. So we've been using guidance from the ECNP app, which has been quite useful. And these categories are based on the mode of action and or pharmacological domain. So antipsychotics as a traditional category has been grouped into primarily dopamine receptor antagonist, dopamine and additional serotonin antagonism, and a partial dopamine antagonist. Antidepressants then have been converted into a serotonin reuptake inhibitor, and also serotonin and norepinephrine reuptake inhibitor. Antiepileptics then have been split into two different NBN categories: glutamate voltage-gated sodium channel blocker and a glutamate voltage-gated calcium channel blocker. So yeah, again, we very much welcome any suggestions, feedback, or questions based on what we're doing here. There has been some and there will be continued kind of updates based on these categories as we invite experts in the area as well to contribute and give their input on this. Thank you very much for listening to me, and all the best.
Hello, I am Joaquim Radua from EDVAPS Barcelona, and I want to share a question. Can we use brain anatomy to predict the individual response to a medication for bipolar disorder? In other words, imagine that a patient undergoes a brain MRI, and the computer uses this image to estimate which medication is more likely to help this person. Well, why are we interested in predicting the response to specific medications? Many classic imaging studies describe brain differences with controls. They show, for instance, which brain regions are increased or decreased in individuals with a disorder. These studies create knowledge, beneficial for future treatments, but not for current patients. Using MRI to predict the response to specific drugs could help them now. The physician would have individualized information about the likelihood of response to each medication group. And this is relevant in bipolar disorder because of the heterogeneity between episodes and individuals. And why do we need machine learning for that? Indeed, we do not need it. Classic tools such as the logistic regression can be used to predict. However, they show overfitting and collinearity problems when using many imaging regressors, for instance, when using the volumes of all brain regions. Conversely, machine learning plus validation in a test set overcomes these statistical issues. Using a test set means that the individuals used to validate whether the model predicts or not are different from those individuals used to create the prediction model. In this regard, I would like to comment on 3 aims I think machine learning studies should have to provide valuable tools in psychiatry. First, studies should aim to avoid bias. With the machine learning hype, maybe we forgot rigor. For instance, classic imaging studies were very cautious about combining data from different sites. Conversely, in novel machine learning studies, analysts sometimes forget about it. Unfortunately, we have previously showed that when using different sites, prediction accuracy may be inflated. Second, studies should aim to be reproducible. Some software tries many algorithms until one predicts. However, this practice is very similar to traditional fishing. If you feed many models, some of them may randomly seem to predict, but such predictions are only lucky guesses. These falsely accurate models will not predict when using new data. Finally, studies should tend to be useful. For instance, do we need a model that uses brain data to predict the sex? That said, I acknowledge that many studies may be indirectly helpful. For example, some machine learning studies were proofs of concept. But the question is, can we predict future outcomes? Of course, it's unlikely that we can predict them very accurately, because they may depend on things that have not happened yet. For instance, the symptoms may worsen after starting a medication, not because of the medication, but because the patient faces an unexpected stressful life event. That said, the idea is that we could use the patient information we have now to predict whether he or she will improve with the medication in the future. I think this aim is excellent. However, it does not seem to fit typical imaging studies, which are small and cross-sectional. They have no information about future outcomes, such as the response to medication, and their sample size is small for machine learning algorithms. The ideal study would be enormous, longitudinal, and possibly include randomization of different medications. But unfortunately, this is extremely costly and unlikely to be available soon. So, can we do something in the meantime? Well, with the ENIGMA Bipolar Medication and Clinical Phenotypes project, we may have extensive MRI medication data that could provide clues about how to predict the response to medication based on 3 assumptions. First, we may assume a two-way relationship between medication and neuroanatomy. Medications change brain anatomy, but there may be also latent factors associated with both the response to the medication and the brain anatomy. This means that individuals likely to respond to a drug may have different brain MRIs. Second, we may assume that the effects of a medication depend on the cumulative dose. This assumption is critical because if we can model a linear relationship between cumulative dose and brain effects, we can isolate the data. Finally, we may assume that by trial and error, patients receive a medication they respond to. The idea is that if a patient responds to a drug, he or she will continue taking the drug. In contrast, if a patient does not respond to a drug, he or she will discontinue it. This would mean that patients that are currently receiving a medication respond more to this medication than patients that are not currently receiving this medication. Here I show the assumption that the larger the cumulative dose of a medication, the larger its brain effects. And here I show the assumption that there are latent factors associated with both the brain features and the response to a drug, and thus with the likelihood of receiving it. Therefore, we would observe that the more intense the brain features, the more likely the patient receives the medication. With these assumptions in mind, we could now do the trick. First, we should find the cumulative effects of medications on the brain using multiple regressions, one for each brain region, and reverse these effects. With this step, we would have imaging data in which we have reversed the effects of medication. Afterward, we could use machine learning to predict the current use of a medication group from this MRI data. This would be analogous to logistic regression, one model per medication group. And according to our assumptions, if we predict use, we predict response. I want to clearly acknowledge some limitations of this approach. First and most important, the evidence provided by this study will be indirect. This study will only provide clues. Second, even if using the extensive ENIGMA bipolar medication data, we will have not enough data for predicting the response to specific drugs, so we will use medication groups. Finally, I must acknowledge that the likelihood of receiving a medication depends on whether the medication improves or prevents the symptoms, but also on whether the medication does not cause significant side effects. This study will mix efficacy and the absence of side effects, though this may not be so important at the practical level. I must sincerely thank all participants in the studies and other researchers from the ENIGMA Bipolar group. Without them, this would not be possible. And that's all on my end. Thank you very much for attending.